more space, is to deal with what's called the central dogma. Which is when Francis Crick of Watson and Crick proposed this, there was not a whole lot of evidence for it. So it was a thing that you kind of took on faith. It would have been in the late 50s, early 60s. And then over the course of the 1960s, this got worked out and turned out to be actually true. So it's now no longer something you have to accept dogmatically, but in fact, there's tons of evidence for it. So basically, the question was, how does DNA, or I'm going to put it this way, a gene which is made of DNA make a protein? So Watson and Crick and those folks who worked with them knew genes were important. They made proteins. Previous evidence had showed that. It was not at all clear how this worked. And the answer will turn out to be base pairing, right? As we'll see. That's how this process works. And it's a rather step-by-step thing that we'll take you through. So I want to give you an overview, and then we'll get into details. So you've got a gene. You start with a gene, and you end up with a protein. And this is information flow. That is copying and translation, but not chemical conversion. So I want to be really clear about this. The problem with this course, one of the many problems with this course, is I use arrows for all sorts of things. This arrow up there means information flows. So when I say gene makes protein, it's not that I'm taking a gene and I'm converting it into a protein. Like sometimes those arrows, like glucose is converted into pyruvate. I'm not converting a gene into a protein. I'm using the information in a gene to assemble a protein. It's like you got a bunch of directions for making some gadget, Lego project or like, and then they use that information to construct the project. So the gene is information that is copied and translated to make the protein. And so there are a couple of steps. So genes are made of DNA, and this is permanent in cells. And DNA is first copied into what's called mRNA, that is messenger RNA. And this is temporary. Talk a little bit more about that later. And then that mRNA is used to make protein, which is also temporary. All proteins last for a certain amount of time in cells and then degraded and reused. And the protein here does the biochemical functions. Things like, eye color, PKU, etc. All right. So there's a couple of steps I want to talk about here. This first step is called transcription. And that is making mRNA copy, or making a mRNA copy of a DNA template. Okay. The second step is called translation, and that is making a protein using mRNA template. And those two terms up there, transcription and translation, are unfortunate in biology. They sound a lot the same. It's like when you go to the ice cream store, plain cone or sugar cone. Well, both of them are plain and both of them have sugar. So how do you know which one's which, right? That's a terribly non-descriptive name. These are slightly more descriptive, but it will take you a while. It wasn't until I was like a sophomore or junior in college before I could say translation, and I knew immediately which one it was. It's like any language, you got to use it, right? Many times until you get it solid. Transcription is making an RNA copy. It's like copying something over, like you're transcribing something. You're looking at some notes and rewriting them. It's a simple process. More or less the same language to the other. Translation is a more dramatic change, changing from one language to another in the normal meaning of the word. In this case, you're changing from RNA to protein. It's much more dramatic. So the way I think about it is transcription, just copying over, is the simple one. Translation is the complicated one. That's the way I originally memorized it. Now it's sort of sunk in a variety of different ways. But we're going to talk about these step by step. The other one, which we talked about last time, is replication. And that's copying DNA for mitosis and meiosis. So copying the whole thing. All right. Now, I'll give people a chance to write that down. And then what I want to show is, if you could turn to the next page of today's information packed handout, there's some still frames from some animations that I want to show you. First one is an overview of the central dogma, and then we'll get into details of the first step. All right. So what do we got here? Let's do this one. All right. So here, I did not do these animations. They were from some folks at UMass Amherst. What I want to show you is just a general overview of the central dogma, and then we'll get into details, like I said. So this is DNA, backbone, base pairs, different colors for A, G, C, and T. DNA is always double. So what we're looking at, we're zooming in on, is that chromosome. So remember, your popsicle stick. Zoom in on it, turn it sideways, and zoom in on it. This is a tiny little piece of one gene. Right? The chromosome extends enormously in either direction. Chromosomes can be 100 million base pairs long. This is probably a couple dozen, so it's a million times longer than this on either direction. A tiny little piece of a chromosome. Imagine it's part of a gene. Our job is to make a protein. So we do several steps, all of which involve base pairing. So here, you start off. You're going to do base pairing. You've got to have some bases exposed for pairing. So the first thing you do is you peel off one DNA strand. This animation shows that strand disappearing. That's not how things really work in real life. That strand kind of goes out of the way, but it's not lost. So now you have some DNA with sticking out bases ready to base pair. Your next job, as I showed on that list, is you're going to make RNA. So you're going to polymerize RNA with an enzyme called RNA polymerase. And how does it know which base to put in? The answer to all questions in this section of the course is base pairing. So only the right base will pair here. And so what happens is the RNA gets made one base at a time. The base basically base pairs and joins, pairs and joins, pairs and joins, and you grow this blue strand, which is indicating the messenger RNA. And so it's the complement of the sequence up here. Right? It's not A and A, it's A for T, etc. So you make your RNA, and then you peel the RNA off, because you're going to use that to make protein. So again, let me stop this for a second. The arrow said DNA to RNA. So you notice we didn't convert DNA to RNA. We used DNA and we read it. We used the information in DNA to make a new strand of RNA. And the DNA is unaffected by this. Now, this animation also shows that the DNA went away. What really happened to the DNA is those two strands came back together, base paired, and that DNA is ready to get read again or whatever. It's back exactly to the way it started. The RNA comes off, and our next job is to make proteins. And there, it also works by base pairing, but they're read in threes, and these simulate the amino acids of the protein that's going to be made. And so you go, you read protein in groups of three. We'll talk about that more later in the week. And so then you make a protein, and then that protein folds up and has a particular structure. All right? So just one last time quickly through. So you start with DNA, double-stranded, part of a gene. You unzip it to expose bases to allow for base pairing. And the thing about transcription, making RNA, is that you only make one strand. Because your goal is not to copy every single gene, but just make the protein that cell needs at that moment. It only needs some proteins at any given time. So you're going to make a transcript. You're going to read it. And the way this works is base pair and join, base pair and join, base pair and join. And you make an RNA copy. The RNA copy peels off, and the DNA goes back to the way it was, double-stranded chromosome ready to be read again for another RNA. So you peel off the RNA, a single strand of RNA from not the whole chromosome, but just a piece of it, just the gene you care about, and you translate it into protein. Questions about that big overview before we talk details? Yeah? So is the other DNA that peeled off, is that also transcribed? It would make sense for that to be true, sort of economically speaking. It turns out almost never. If you work out what the sequences would be, it's very, very hard to have a sequence that codes for a protein reading one way and for a protein reading the opposite direction on the other strand. So it almost never happens that way. Basically, one strand gets read and that's it, and the other one is ignored. Other questions? Yeah? Does it turn into? Let me talk about the details of that in a second. So I want to talk about details. There was a question about the details of transcription, and I'll talk about that actually next. All right. So let me show you. Let's go into some detail. All right. So when we're going to talk about transcription, let me put it this way. Do it in black. Right? So that's DNA to RNA. So the question is, what's the differences? What is DNA versus RNA, the chemical differences? So I want to say a little bit about that. If I look at DNA, and I want to show you one nucleotide in a chain, I want to sort of zoom in, all right? And DNA's goal, by the way, is its, well, let me put that down in a second. All right, so let me get myself a little more space for this diagram. In a chain, there's going to be the previous nucleotide. And you guys saw some of this when you in the pre-lecture quiz for last week's lecture. There's a phosphate group, CH2, and then there were these rings. This was drawn very schematically in the pictures on the web. There's a sugar, and there's a base hanging off of the sugar. And then there was an O, and then the next nucleotide. And there's a couple things that I can explain here. There's a numbering system. Where do the primes come from? This thing here is the sugar, and the atoms on the sugar are numbered: 1 prime, 2 prime, 3 prime, 4 prime, 5 prime. Okay. Why are they numbered like that? Because if you want to talk about their chemistry, it's actually a little bit of chemical nomenclature. If you take organic chemistry, this kind of thing will matter. If I want to say, as we'll see in a second, this sugar is called 2' deoxyribose. Bless you. What that means is it's ribose where the 2' oxy has been removed, deoxy. So deoxyribose means somewhere in that sugar molecule I've taken an oxygen out, deoxy. How do I know which of the many oxygens in the molecule I've taken out? I've taken the one off of the 2' carbon. You're never going to need to know these for this class, but just because lots of times people wonder, what does 5' and 3' mean? Where does it come from? You number those carbons. It's essentially arbitrary. You could number them going around the other way. You could start at a different place. It's a convention of chemistry that when you have 2 rings joined, one ring is numbered 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, like the base. Those are numbered 1, 2, 3, 4, 5, 6, 7, 8, 9, whatever. The sugar, the second ring, is numbered 1', 2', 3'. If there was another ring, it would be 1', 2', 3', etc. So the 3' carbon is the one on the ring. The 5' is the one that sticks out, if you remember that from the questions for the before lecture quiz. So a couple of other details about these guys. The base is A, G, C, or T, and A pairs with the T, and G pairs with C. All right. The other thing about DNA that's important to know is that DNA is used for information. It's for information storage, and it breaks down slowly. It turns out, for chemical reasons, because it doesn't have that 2', there's an oxygen missing there, DNA molecules are much more stable than RNA molecules. And they've, in fact, isolated DNA molecules from woolly mammoths from 40,000 or 100,000 years ago. The DNA is highly fragmented, breaks in the backbone, but it can stay intact for enormously long periods of time. And it turns out that's an advantage because the purpose of DNA is to store information. You don't want it breaking down on you. So that's what a DNA nucleotide looks like. And if you compare that to an RNA nucleotide, so one nucleotide in a chain, they're very similar, right? It turns out there you've got the previous nucleotide, and you have this sugar phosphate, or this phosphate sugar, if you like, backbone. You have this 5-carbon sugar with bases hanging off. And you have the big differences here are that here's the O to the next nucleotide. And if you look around the ring, there's an OH, and that's the difference between these two. If you number these guys, 1', 2', 4', 5', the sugar is ribose. And this thing here, this OH, is not present in the DNA. And it turns out that the 2' OH makes this less stable. Therefore, a temporary working copy. That is, these molecules work the way they do because it turns out there's some chemistry of that little OH there that's not present in DNA that makes RNA much less stable. And so it tends to fall apart, and you have to keep making it new. And the reason for that is I don't want to be making all proteins all the time. And in fact, once I've made it, I might not want to make it again for a while. So you want to make the RNA go away. So RNA is a temporary copy. And the bases in RNA are A, G, C, and U. And A pairs with U, or T sometimes if you're going to pair with DNA, and G pairs with C. All right. Questions about that chemistry stuff before we talk about how transcription actually works? Yeah. Oh, yeah, why is there U instead of T? It isn't clear. I haven't read about this in a long time. It's just one of those things it is. I don't really know. I'm not sure that there is actually a reason. Some of it just is. It happens to have evolved that way. Other questions? All right. So what I want to talk about now is actually some of the details of transcription. That is a reminder, making an mRNA copy of DNA so protein can be made. And so that's the goal of this. So we're going from DNA to RNA. So in DNA, there are 4 letters. That is A, G, C, and T. In RNA, there are 4 letters. A, G, C, and U. So the conversion is pretty simple. DNA is 2 strands. RNA is 1 strand. Right? And this is done by an enzyme called RNA polymerase, or RNA Pol for short. Not a super imaginative name, but very descriptive. It polymerizes RNA. It makes polymers of RNA. And a couple of things. The note here is that only one, or so I'll say therefore, only one DNA strand is read, right? Which is the first sort of odd thing, but true. And the other thing to remember is that only the part of the chromosome corresponding to the gene desired is transcribed. That is, again, every cell in your body has all the same genes. You just don't need them all at the same time and in the same place. So any given cell is only making a subset of the possible proteins you care about. So you're only transcribing the genes that you care about. So let me draw a schematic of this and then go into final level of detail. If you have a chromosome, it's 2 DNA strands that are base paired. Right? And if you look at one gene, so the chromosome is a long piece of DNA. And I'm not going to draw them all in. There's zillions of base pairs, thousands at least. And it extends off in both directions. Okay? At one end of the gene, you have one signal sequence. And it's usually drawn with an arrow. This is the start transcription signal. This is a DNA sequence that is recognized by RNA polymerase. It's called a promoter. And in gene X, it's TATAA, ATATT. That's its sequence. So let me back up and explain a bunch of pieces here. If the point is to only read part of the chromosome, RNA polymerase is supposed to make a copy of a part of the chromosome. How does it know which part to make a copy of? You got to tell it what to do. Right? And you also have to remember how molecules work. They can't see in the distance. They can't think. They don't have memory. They just bumble around. So what you have to do is you make a sequence in the DNA that RNA polymerase will recognize. How does it recognize it? Hydrogen bonds, ionic bonds, the whole 9 yards. Basically, the RNA polymerase bumbles around the cell. If it bumps into DNA, it goes, what's that sequence? Oh, that's not a promoter. Never mind. Finds another part of the DNA, grabs on it, goes, oh, that's not a promoter. But if it happens to find, like in gene X, a simulated situation, finds TATAA, it goes, oh, that's the right place. I'm going to start making RNA. Sequences in real organisms are typically longer than just 5 nucleotides. But that's the basic principle. There's a sequence of DNA, particular bases, TATAA, GCC, AAA, whatever, some particular sequence that RNA polymerase has evolved to recognize. And once it sees that, it grabs on and starts making RNA. And that's how it, and so that's also, so for example, if you were trying to get any RNA, unless you have this sequence in your gene somewhere, you would not get any RNA. And if you had this sequence and you messed it up, you changed it, then you will not get any RNA because you haven't told RNA polymerase where to start. Right? So that's the signal to start. And what happens then is the RNA starts getting made, and the RNA goes along, and the 5' end is what corresponds to the promoter, mRNA, and this is one strand. Okay, and you've got the same deal, right? You've got a RNA polymerase. You don't want it to keep going forever. You only want it to read this gene, so you got to tell it when to stop. Again, RNA polymerase doesn't know anything, doesn't have any foresight, doesn't have any smarts. It's going to have to be a DNA sequence. So there'll be if you want it to stop at the end of the gene, you have to put a signal to tell RNA polymerase to knock it off already. You've made enough RNA. So there's a sequence in the DNA. The stop transcription signal. It's called a terminator. Okay. And in GeneX, it's the G, 5 Gs over 5 Cs. Okay. In real life, the signal is a bit more complicated than that, usually longer. But GeneX is just for simplified purposes. But it is a particular DNA sequence. When RNA polymerase gets there, it goes, OK, you told me to stop. I'm going to stop making RNA. And when RNA polymerase gets there, that's where it puts the 3' end. That's the last nucleotide it adds, and that's it. Okay? All right. Before I talk about particular details, which strand, how you figure all that stuff out, I want to make sure everybody's with me with promoters and terminators and starting and stopping RNA. So that's what you would have seen, would probably have driven you crazy. That green and red block indicated the start and stop transcription signals. And you have, although you don't really need a stop, once you get started, you can make some sort of a strange RNA. But you got to have a start, otherwise you can't make any RNA at all. RNA polymerase will ignore your DNA. Questions about that stuff? Oh, yeah. Are those the only letters that work as a promoter? In GeneX, yes. In real life, it's a longer sequence, and there's some slop allowed. Like, for example, TACAA might also work. Okay? In real life, it's a little more complicated. And like I said, longer sequences typically 30 nucleotides long, but that's just a lot to write. But it is a particular sequence. Yes? So the U's, if you saw U's, they would have been in the RNA sequence. Right. Other questions? Yeah. Do the signals get so do the signals get translated? I would fuss about terminology. Do the signals get transcribed? And the answer is no. The signals are not transcribed. So promoter and terminator, actually that's a good, I should make a note about that. Promoter and the terminator are not transcribed. All right. So let's talk details. So it's a step-by-step process. Number one, RNA polymerase binds to the promoter DNA. How does it know where to bind? It doesn't know where to bind. If it tries to bind to the wrong place, the hydrogen bonds and the shape is wrong. It just doesn't stick. It only sticks by hydrogen bonds, ionic bonds, blah, 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 if the shape is right. And that means if the sequence is right. So it binds to promoter DNA. And then, 2, RNA polymerase unzips some DNA, breaking the base pair, separating the base pairs. 3, RNA pol starts making mRNA, and always 5' to 3'. That is, you add to a 3' end. Therefore, you read what's called the transcribed strand 3' to 5'. Wait, how come it's backwards? If you're making RNA one way, base pair, the other strand has to be going the opposite direction. In order to form proper base pairs, the two strands have to be anti-parallel. I'll draw a diagram in a second that should make that, I think, a bit clearer. So let me draw a big picture of this. So here, 5' end of your chromosome, dot, dot, dot. I'm going to draw what's called a transcription bubble. To 3' end, big fat transcription bubble. Base pairs here, 3' and 5'. So this is the DNA. This is also the DNA. Right? And if we draw in color, here's the promoter. So it says make RNA. So make RNA going this way. So promoter has, not only does a promoter say start, it says start and go in a particular direction. So what I want to do is put some bases in, and I want to have you guys think about what RNA gets made. So say the DNA, just for the sake of argument, is T, A, C, G, C. Let's just say. And down here, what base goes down here? A, T, G, C, G. Cool. All right. So I'm going to make RNA. I think I used blue for RNA. Yeah, I did. Blue for RNA. I'm going to make RNA go in this way. All right. What I want you to do is talk to your neighbor and figure this one out. Which of the RNA, which end is 5', which end is 3'? That's the first thing to do. Then, which strand is that going to be base paired with, top or bottom? And finally, what's going to be the sequence of the RNA? And the secret to this is there's only one RNA you can make that follows all the rules. You might imagine there are two, but there's only one that fits the rules. So think it through. The best way to start is which ends 5, which ends 3, what's it going to pair with, what's the sequence going to be, and then you'll be all set. Right? But so like to say, there's one RNA you can make and only one. So think about it, talk to your neighbor, and I'll call names in a minute or so.